We're going to move on to our next speaker, who's Dr. Rosalind Bernstein-Mannon, who is a personal and I think for the entire kidney transplant world, inspiring role model. She's a professor of medicine, pathology, and microbiology at the University of Nebraska Medical Center, also vice chair for academic development and research mentoring, and associate chief of nephrology for research. She's a past president of the American Society of Transplantation and deputy editor for the American Journal of Transplant. She received her MD from Duke University, completing internal medicine, residency, nephrology fellowship, and chief residency at Duke. Her career includes serving as medical director for the Kidney Pancreas NIDDK Intramural Transplant Program and at the Birmingham VA Medical Center, and section chief of transplant at the University of Alabama at Birmingham. She is the chair of several committees and initiatives, including Women in Transplantation, chair of the Policy and Advocacy Committee of the ASN and Scientific Registry of Transplant Recipients Review Committee. Her lab research focuses on mechanisms of chronic allograft injury using in vitro and rodent models, and is funded by the Veterans Administration Merit Award. She's authored numerous publications on all things kidney transplant, including mechanisms of chronic allograft failure following transplant, post-transplant complications, therapeutics, and immune monitoring. She's also a self-proclaimed lacrosse maniac. Today, she'll be speaking to us about unmet therapeutic needs. What are they, and can we find the fixes? Welcome, Dr. Mannon.
Thanks.
And the Oregon Ducks, sorry about that, forgot to mention.
I know, well, so the Oregon Ducks women's lacrosse program was closed down last week due to COVID. So the Oregon COVID rates are high, and five players from 3 households were infected, including one in the dormitory. So that was a very abrupt end to our season. And then my daughter's old team is going to be cut for this year. So they have one more game today. They actually won their playoff, shockingly. So thank you all. I'm taking up some of my important time to chit chat with you, but John Arthur, I forgot about you being one of my interns and how we would round at night as the chief resident and get into trouble. But John also failed to mention the many hours at the lab bench where I'd be suffering trying to do pH measurements in T cells. And also, John, I appreciate your comments. I got very emotional this morning listening to NPR and about what's happening in India. And it's overwhelming to me as a human being, and I hope that some way we'll get through this. So today my topic is very different from the other topics. Dr. Farouk asked me to talk about unmet therapeutic needs in transplant and what are they, and can there be fixes? And so this will never be on a board exam, I can assure you. These are my conflicts of interest, and I'll be discussing off-label use only in the context of regulation. Over the next few minutes, what I'd like to do is review the progression of immunosuppressive drug development that's occurred over the last couple of decades, highlight the barriers for clinical investigation and innovation in transplant, and then educate you as, to the public to understand a recent initiative with the Food and Drug Administration to advance transplant therapeutics. So quite simply, this is one slide that explains and highlights all the unmet needs we have in kidney transplant drug therapy. We need to be better addressing death-censored graft failure with therapeutics that are effective, particularly applications such as antibody-mediated and chronic active antibody-mediated rejection, delayed graft function, and BK virus nephropathy. There are no approved therapies, and they continue to be quite difficult for us to deal with. We need to be able to assess the sufficiency of immunosuppressive therapy for each patient. And I have been challenged by this my entire career. It gets a little bit better because I can look at patients well. That was part of my Duke training. You could sort of figure out who was sick just by looking at them, believe it or not. It's a skill, but we rely primarily on renal function, as you know. And we have no approved biomarkers for efficacy. We measure drug levels and we do biopsies, and that's kind of the way we know what's going on. And measuring donor-specific antibody. We need to be more effective at managing the increased risks of cardiovascular disease. One of the key causes of late graft loss is death with function. So when you look at the 50% of individuals out more than 11 years, half of them will lose their graft because they die. And it's not a surprise when you look at our patient population, hypertension, diabetes, dyslipidemia, and obesity. And these issues are aggravated by current immunosuppressive therapy. And we also need to have therapies that really promote patient adherence, but also physician adherence, that physicians feel comfortable maintaining a certain level of immunosuppression in terms of long-term outcome. So I would say that I was very inspired. I went to the NIH around 2000. I was very excited to be there because we were primarily a program looking to investigate new novel therapies. And this is a summary of those in 2002 review by Flavio Vincenti, highlighting biological therapies, T-cell activation therapies, and lymphocyte migration. And our program was on the ground floor of several of these biologic agents in phase 1 and phase 2. And the review highlighted that these medications meant new opportunities, more stable long-term graft function, less rejection, knowing that rejection at that time was about 8 to 10% rate per year, just as it is now, and the avoidance of calcineurin inhibitors, the avoidance of a long-term nephrotoxic agent. But reality set in about 5 years later. This was published in 2008. Flavio teamed up with Alan Kirk, who was then my colleague, that indicated that the first part of the millennium had been very disappointing. And again, when I compared it to when I was a fellow and a medical student, there was really no therapeutic innovation, that all the immunosuppressants on the previous slide that we showed were not approved yet, that there were still some agents in trial, particularly CP690-550, which is a JAK inhibitor, and belatacept, which you may recall and we'll see has been approved. But things like eculizumab and alefacept were in trials, but I will tell you, failed to go further. And so the pipeline really became what he would show as this sort of cemetery for many of these agents. And the issues are summarized here, that the success of existing immunosuppressive regimens hinder the future development of drug therapy because of the vanishing primary endpoint of acute rejection. That rejection rates are so low, it's hard to cut that down even lower without a massive trial. And the difficulty in challenging demonstration of improvement in long-term graft function. It's very hard to show changes at 10 years because you can't have a trial for 10 years. We can't wait for 10 years. There has to be a way. And so when you look at recent new drug applications, and I must say, starting to look back at 2000, that was 20 years ago, but that turn of the century of the millennia, we had Rapamune or an mTOR inhibitor. 10 years later, it took to get another mTOR inhibitor. There's nothing magical here, but a dearth of any approved therapies. And then a year later, belatacept was approved as a novel agent. And that's pretty much been it for 2 decades. You can see that the last few years, it has been congeners of tacrolimus for long-acting agents, and Varsa's getting 2 because one was a conversion and now there's a de novo application. So very disappointing. So when you all are talking about networking, I think that is so important. And when I was the incoming president of AST, American Society of Transplant, somehow I got invited to some dinner with the American Society of Nephrology. Then president was Ron Falk, and he sat me down in this very nice French restaurant in Washington, D.C. So he was on my left and Joe Bonaventure was on my right. So what could you say to that? And sometimes networking is not about making an impression like, oh, that Roz Manin, she's great. It's listening to what they're doing and getting ideas. And that's really when I network now, a lot of what I spend my time doing. And Ron was just like going on and on and on about this thing he called the Kidney Health Initiative. Now, some of you know what KEI is, or KHI. It is a private-public partnership with the Food and Drug Administration. And Ron kept going on during dinner about this therapeutic lack of innovation in nephrology that many of you are familiar with. And he felt that this would overcome a barrier. It would put all these groups together with the Food and Drug Administration. And the Food and Drug Administration had specific interest in this. They actually have guidance online shown here where they partner with experts to attack specific public health needs and bridge the scientific gaps. And he pointed out 2 private-public partnerships that I was not super familiar with, but now I am. Critical Path Initiative, which we'll talk about momentarily, and the one I should have known because I was at Duke forever, the Duke DCRI Cardiac Safety Initiative. This initiative really focuses on adverse events and all of those massive cardiac therapeutic trials. And so leveraging and the ability to gather data and review it in a public and partnership with FDA is really quite important. And so Ron went on to achieve his dream, and he really leveraged mine. So when I was president, I really made this my sort of background. I mean, you're working on all these other issues. There were all kinds of crazy things going on. We had to worry about the HOPE Act, the National Living Donor Assistance Program was in trouble. The CDC had these high-risk guidelines. And, could you imagine telling a patient you might get HIV? Well, it was very difficult. And so, but amongst that, in the background, I spoke to the FDA. They really felt that transplant should stay with KHI. They weren't interested, I could tell, in another private-public partnership. And they said, well, draft a document, put together what are the unmet needs, put a white paper together. And white papers can be very complicated. It takes like a lot of people and a lot of meetings, and we didn't have time. So I drafted the people I know that could do the job. My colleague, Rita Alloway, a PharmD at University of Cincinnati, a well-known clinical trialist. Tim Schroeder, past surgeon in transplant, a scientist in transplant, but then became the CEO now of one of the largest clinical oversight organizations known as a Clinical Trials Institute, then mostly focused on transplant, now universally and internationally. And my colleague Flavia Vincente, probably the expert clinical trialist of my time in transplantation. And we drafted this almost 150-page document called the Call to Action. So what's in this paper? You can look it up on the PDF. We never actually tried to publish it because I knew peer review would just tear it apart. But so we set several arguments to the FDA for their interest. One, that there were these unmet therapeutic needs in all of solid organ, not just in the kidney population, but again, improving long-term patient survival after all transplants, improving the function. And we even threw things like small bowel, composite tissue, and xeno. I mean, you're going to go with the gusto. Improving the number of donors and improved function of non-ideal. So you saw that Klaus Niemann paper. A lot of devices and pumps and new solutions were coming out. And that included things like stem cells, immunosuppressive medicines that minimize comorbidity and improve quality of life, that are maybe more specific by avoiding the induction of viral diseases and cancers. And finally, specific therapeutic indications like antibody-mediated rejection and delayed graft function, we even put intolerance and recurrent disease. So we really threw the money in here to get their attention. And most importantly, the single biggest argument we had was the misconception that transplant outcomes were outstanding and could not be improved upon. And I have to say that this was probably one of the biggest arguments that they did not understand. And how did this come about? A colleague of mine, Randy Morris, who was a co-author in an upcoming paper, was a cardiac surgeon, transplant surgeon, and had a huge lab. And he made this argument over and over again. He said, why aren't we treating kidneys like oncology, this therapeutics like oncology? And so we pulled a bunch of data from USRDS and from one of the big cancer registries. And when you look at patient survival at 5 years, yes, live donors do great. Deceased donors a little bit worse. Waitless mortality is tricky. And obviously that's why we transplant patients. We get them off dialysis. But when your transplant fails and you go back to the list, your mortality is even higher. And so if you summarize all of these different population survivals, you can see here a mean survival at 5 years. And on the right, you can see some cancers. I mean, these are regional breast cancer and regional colon cancer. I mean, I never, I was on this path to be an oncologist, and I said, it's just a lot of it is just too much. I want to be taking care of living people. And this slide summarizes that we are like oncology. We have to really pay attention to what's going on. And I'll show you here, this is rejection rates shown here for all solid organs, but we highlighted the low rejection rates, which is a success story. But that long-term survivals at 10 years needed to improve. I shouldn't have to say to a patient, you're getting a deceased donor and I'll see you in 10 years for another one, or on the average 10 years. But this is the hard truth. And it's even worse for some of the other organs where there's no fail-safe of dialysis. We also emphasize patient-reported outcome measures of the side effects. These are things that I think I probably take for granted, although they take up a lot of my post-transplant clinic time. Things like diarrhea, headaches, tremor, difficulty thinking, and importantly, pill burden. We just think of the immunosuppression, but then you look on this 2-page medication list and you realize people are taking 60 or 70 pills a day, every day. I can't even take a vitamin every day. I don't know about you. Diabetes, hypertension, often aggravated by medications, and then the faithful return to dialysis. So Alice and Tong worked together with a number of you all that are listening today, and we created a standardized outcome in nephrology and kidney transplantation to really guide us in terms of what are the outcomes patients are most concerned about when we're designing clinical trials. So I attached this big document the day before my presidency ended. I put it in an envelope and I FedExed it to the NIH, and I outlined really sort of a background about transplant, these unmet needs, and our approach to a therapeutic and device development, which involved a private-public partnership with them. Now, after you become president, they sort of put you in the, in the mothball bin, and you just kind of hang out and say, oh, I think that sounds like a great idea, or don't do that. I tried that, it didn't work. Good luck. So about 3 months later, the Office of Translational Science, which is the Office of Food and Drug Administration that is supposed to promote innovation in drug regulatory reviews, met with me and another colleague from the American Society of Transplant Surgeons, and really was intrigued by this and recognized that we had made a good argument. I'd also done a meeting at FDA in their division, in the transplant division, that was really infectious disease drugs, and I got up for 10 minutes and just said, there's a problem in transplant, here it is. And so I had grabbed some attention. So this wasn't exactly like a bomb that I threw into their in their mailbox, but these were 3 individuals they had recommended we consider that we should talk about private-public partnerships with, including Arthur Holden. We spent quite a bit of time with him, probably too much time because he was just too focused on adverse events, critical path, and then the Biomarker Consortium. And so really, in the end, we finally settled by working with Critical Path. A Critical Path known as CPath was founded in 2005. It's an independent nonprofit. It is basically a large private group that makes public partnerships with FDA, and they have all different subsections and consortia, as I'll show you, and they have an opportunity to bring all this expertise together. To keep the pressure on, we continue to have meetings. We had an FDA workshop on endpoints for transplant trials. We met in September 2015. We had a follow-up meeting with the Transplantation Society, so we got international involvement in this effort to look at endpoints, to create new trials. This is a paper summarizing those findings. Again, reminding us that oncology had done, while we were sitting and getting 1 drug approval, oncology had approved about 41 drugs by what's called surrogate endpoints for approval or rapid drug approval, and we'll go into this in detail. And the surrogate endpoints that are used in oncology, like a lot of these drugs that we hear about on TV, are disease-free survival or objective response rate or progression-free survival. And I have to be a little bit, just to let you know that while this has always been my mantra, oh, let's be like oncology, there was a great perspective last week in the New England Journal called Dangling Accelerated Approvals. When you do an accelerated approval, much as oncology, then you have to do a hard endpoints outcome study to confirm these studies. And so this slide says 26 of 35, that was from 2015. This gives a great review on the PD-1, PD-L1 inhibitors and how so many have yet to receive full FDA approval. And we need to know about it because of the AKI association. We had another viewpoint, again, highlighting the barriers. And this is maybe a little bit easier paper to digest with, again, my colleague Rita Alloway, Randy Morris, who I mentioned, and Mark Stiegel from the ASTF. Again, what was stopping us, the field? Well, there was this misconception that we couldn't get better. I think many of my colleagues really were saying, well, I don't see, they weren't seeing these patients at 5 years, 10 years, 12 years where they had issues. There were current regulatory barriers that the FDA for transplant was very focused on an endpoint of graft rejection and patient and graft survival. The industry was not excited about us. They took all their new things into patients with psoriasis. Why do you think that? Well, a psoriatic patient has one thing, and they have skin lesions. It's very easy to monitor. You don't need a biopsy. You don't need a ton of blood. The side effects are just the side effects. But transplant patients, if you came into your clinic, the average adverse event of a standard patient in transplant, it's like tons and tons of things wrong. Every complaint is always a significant issue. And so I think they had lost interest in this small field that had complicated patients, and they were worried about the return on the investment. They would put all this money into these trials, and yet these patients were quite sick, and it was complicated. And so just the diehards in the field hung on into their bigger companies that maintained their interest and focus in a small division in transplant. And finally, we're kind of a risk-averse community. And we just heard a talk about donors, and the big problem in transplantation that's really he didn't really get into in detail is people are worried about bad outcomes. Arkansas's program has amazing outcomes, excellent outcomes. And I think it's because they're just not taking every kidney out of the bucket. But that's kind of the problem that some of the transplant field has, is they become very risk averse. And so, using a new therapy and risking a rejection event and a graft loss is just not their cup of tea. So despite the fact that I had sent that letter to FDA in 2013, it took really 4 years. There was a lot of political behind the scenes I can't share with you. I was somewhat disappointed that the founding members did indeed remain AST and ASTS. I'm located here as a member of the steering committee, but this is our Transplant Therapeutics Consortium. These are societal and government partners, and these are biopharma partners. The list is much longer now. And shown on the right here is all the consortia that CPATH manages. And I didn't have a new logo, but the acute kidney injury, there's a new acute kidney injury consortia focused on developing and advancing biomarkers and therapeutics in AKI. So you need to pay attention to this. Polycystic Kidney Disease Consortium is another consortium that works. And the expertise really exists in CPATH to do this kind of work that I'll show you. They are a private-public partnership. They sort of stand in the middle and bring these groups together. They're in what's called the pre-competitive space. So the industry partners come and they share. It's not like J&J is fighting with, AstraZeneca is fighting Merck about which SGLT2 inhibitor that's there. They may be sitting together and working together to see how diabetes affects long-term outcomes. And the most important thing is, besides having partners together that are really focused in this area, they also particularly have the regulatory experience to negotiate what the FDA would like when you're trying to develop new therapeutic endpoints. And they can do this both through FDA and iterate it through the European Medicines Association and the PMDA, which I'm blanking on the initials of what they stand, but that's the Asian equivalent of FDA. And our Transplant Therapeutic Consortium is focused primarily on new therapies for patients right now. We're not anything else, even though I had a crazy idea. So when you are trying to get a transplant therapeutic approved, by and large, the way it has always been is you have a comparator arm. You don't do a single arm trial. You have a control arm, which was mycophenolate, prednisone, and tacrolimus. And actually, for all the trials up into belatacept, this was always cyclosporin, and this took forever to get changed. This was, again, the project of my colleague, Flava Vincenti, when he was president, is he got tacrolimus changed to the control arm. And the current endpoints are biopsy-proven rejection and patient graft survival. So by definition, any new therapy that is gone in this kind of approval pattern will be non-inferior. You can't show, it'll be very hard to show superiority because you would need like 5,000 transplant patients to show an improvement in graft rejection rates. And moreover, it's kind of unlikely that if the product is just non-inferior, which is the only thing you could do, that someone like you or me would probably say, well, I'll just try that today, what the heck? Because you're comfortable with what you need. And if that product isn't any better, why would you switch to something you don't know how to use? So it really, this kind of drug trial approach would just allow superior therapeutics, which limits any interest in any forward mobility. There has always been this surrogate endpoint accelerated approval called Subpart H in FDA lingo. And in fact, it's really now more popularized because we now have law. We have the 21st Century Cures Act, which was passed in 2015, supported by all the kidney and transplant organizations to support and accelerate therapeutics. It was really never used in transplantation because we didn't have biomarkers to serve as surrogate endpoints for clinical trials. In Subpart H, you have a surrogate endpoint. It's a marker of a drug effect. It doesn't affect how a patient looks, functions, or feels, but it's reasonably likely to predict a clinical benefit. And oncology, HIV therapeutics, hypertension have used this to date. So the first mission, and again, this is another slide that kind of highlights what surrogate endpoints are, highlighting again a barrier that there were no FDA-approved surrogates or EMA-approved surrogates. This is from O'Connell's paper from the Transplantation Society. And some candidates that have been suggested are GFR and proteinuria, which are prognostic but probably not predictive. And it's been tough to get GFR by the FDA. Donor-specific antibody, we talked about this ad infinitum in 2012 and 15, but lab methods aren't standardized. And histology would be a great, but the problem is histologic patterns aren't always disease specific. And a great example is acute tubular necrosis, where you could see that with ischemic injury and a transplant from delayed graft function, but you can also see it associated with antibody-mediated rejection as well. So we have another bunch of other biomarkers that you're familiar with. I'm not sure we'll talk about them in this meeting, but peripheral blood gene expression has been developed and now is a company-based test called TrueGraph, and it actually just has received CMS approval to be used as a monitoring tool in transplant patients. Gene expression with the Allograft has been a long-term project of my colleague Phil Halloran. He created what's called the molecular microscope with all these arrays of gene expression from a biopsy core, and it's called the molecular microscope. And this was just approved by CMS, again, as a potential test assay that you could use in concert with histology. I'm working with a group on gene expression in urinary sediment cells because, as you know, urine is where it's at. And I call urine the bronchoalveolar lavage of kidney transplantation because we have overlooked this fluid for too long. And cell-free donor-derived DNA, which is now 2 commercial assays, Prospera and AlloSure, both approved, CMS approved, meaning that if you order it in the context of the use of, which here is a monitoring use, you can have it reimbursed by Medicare, but these expenses are very high. So it's important to point out with these biomarkers, how are they developed? And so are they developed for clinical decision-making? Were they utilized in the laboratory to identify specific situations? Or are they used for regulatory decision-making? And all the other things I've talked to you about were really focused in academia and also used for clinical decision-making, but not developed as a regulatory decision-making effort. And so I think that this has really affected, it takes a lot to move something from an academic pathway to a regulatory pathway. Some of the assays, most of the assays have analytical validity. They're okay in the lab, they work and they're dependably accurate, but the clinical validity is lacking, and there really has none of those tests has been used in a clinical trial specifically. And many are diagnostic. On that previous slide here, these are mostly diagnostic tools. Is there inflammation, yes or no? Or what kind of inflammation when you're using the biopsy specific, but they're not predictive or prognostic of long-term graft failure. So they're not really going to be helpful in the goal of trying to evolve new therapies, at least not yet. So always the FDA has a process. They like public workshops, and it's great for them. We work in concert with societies and the TTC, and we get all the individuals in a room. And this was a 2-day workshop on the right therapy, the right dose and regimen for the right patient for individualized therapy. It was a 2-day meeting that was all day from 7 a.m. to 6 p.m. It was really focused on day 1 on biomarkers, and the second day, which we won't talk today about, is about patient-reported outcome measures. So we really, to get this meeting to go, we had a lot of pre-education, including myself, because what I knew about biomarkers and how to clinically qualify them was very different than regulatory qualification. And this is a great paper from Chris Leptak. If you're a wonk in this area, if you like clinical therapeutic innovation, this is something to read because it highlights the evidentiary standards that are required for regulators to accept your biomarker, to use it in a clinical trial. And they highlight the differences between a clinical use versus a clinical trial. And importantly, they highlighted groups of biomarkers and types of biomarkers, and that you need a context of use statement. Just like you use a medication and there's like a description of why you would use a medicine, the biomarker has to have a description of how it'd be used. In particular for the FDA, you have to say, I'm proposing to use this test, and this is how we propose to use it. And shown here, this is a table from our paper, were the 8 categories of potential biomarkers. One of the big ones we're looking at is this reasonably likely surrogate endpoint to correlate with long-term outcomes. And it's a big process. I mean, and it's something that, as an individual may be very difficult to do, especially with limited resources, but it would take all your time because you need a letter of intent to the Food and Drug Administration that you're planning to do this. They have to say, okay, they have to first be interested in this. Then they have a review process. Then there's a qualification plan that's submitted that's quite detailed, responding to some of the concerns they've already highlighted by your initial letter of intent, which is already like 30 pages. and then you submit a full qualification package. And I will tell you, for the Transplant Therapeutic Consortium, we've only done step one. We reviewed a lot of different biomarkers. They're summarized in the paper in terms of how they would be used in clinical trials, not their clinical utility. I'd point out two. One is the applet mismatch you've heard about, which was the goal of it was to sort of risk stratify subjects or individuals for trials or for high or low risk pre-transplant. But the one that was the most mature as a reasonably likely surrogate endpoint was the integrative box to predict long-term graft function. So some of you may have heard of the iBOX. It's got a cool name, but the integrative box, it's basically a multi-parameter composite endpoint. It was developed from my colleagues, Alex Lupe and his team from the Parish Transplant Group. It was based on several other markers from a large literature review, about 33, but their cohort included 4,000 patients from 4 centers that had had biopsies in the first year. And it was validated by nearly 4,000 more individuals that were both in Europe and in US. And it had over 1,000 graft failures and almost 8,000 subjects. And they identified through their analytics 8 factors that were associated with long-term outcome, and they called it the iBOX. These include histology, which is inflammation and tubulitis. This is BAMF criteria for BAMF allograft pathology, glomerulitis and peritubular capillaritis, interstitial fibrosis and tubular atrophy, and transplant glomerulopathy. So those scores were important. The development of donor-specific antibody at a set time point in the first year, the presence of proteinuria, and the estimated GFR at the time of biopsy. And they showed 2 things: that the iBOX in this paper could accurately predict graft loss using data from various time points post-transplant, but predominantly the first year, and that iBOX also predicted responses to therapeutic outcomes. This is a neat paper. I would suggest reading it. Shown here are these independent, this is the entire hazard ratios and the number of observations. Note that the donor-specific antibody wasn't uniformly found in all the databases, but any DSA, the lowest level of DSA, and this is right at the borderline for most labs of being positive cross-match, but once you went above that, it had a significant hazard ratio. But our good friends, proteinuria and estimated GFR, have significant impact as well. So the biopsy to me is important, and I love histology, but I also want to point out that the eGFR and proteinuria were important. And then they show 3 therapeutic applications: antibody-mediated injury, T-cell-mediated injury, and minimization trials, and show with the blue bars mean that the iBOX score goes down, and those were correlated with patients' outcomes being better. So again, and they had a C statistic of like 0.81, which is pretty good, but they took different trials and ran this iBOX on those patient data, which is really an undertaking, to say the least. So we decided to adopt it. They were in collaboration and agreed to work with our TTC. And the letter of intent was sent in June 2020. The idea was to use the iBOX in the first year after transplant as a reasonably likely surrogate endpoint of 5-year graft failure for use in clinical trials and the evaluation of immunosuppressive therapy applications that would be submitted through this accelerated approval program. So very upfront that the TTC was going to use this as being the marker, and that led to a very positive response from FDA. That was about 20 pages. And so whenever you get these rebuttals back from your paper, this was nothing like that. This was like leagues beyond that. But again, there were also a team of people. I was involved in part of drafting this letter of intent, but the team at TTC at CPAF really took this over. I'm allowed to show you a snapshot of data. So these are all the trials and participants that have agreed to allow us to use data, and we, meaning TTC, not Rosmanin. But the qualification will include both studies as well as single center registries. So Mayo Clinic has a very large registry. My colleague Ilkka Hankera, Helsinki, has a smaller number of patients, but very nice cohort. Other pharma studies that have been done in the last 2 decades allowed their inclusion, which was great because it's a partnership. I mean, you could never get for them to share this with just an academic center, but individuals felt comfortable doing this. So we have 22,000 kidney transplant patients that we're looking at data, all this information that's available, 2,000 graft failures, 1,500 deaths, and biopsy-proven rejection in 1,600. So plenty of events to look at, and a very diverse population, although I will say that these tend to be quite Caucasian, quite European. There is one database from Houston that has many more African Americans, but it's smaller and it will not be necessarily in the validation cohort. This is a timeline. They actually move things up because things are moving along. I didn't show you the data digest, and we have meetings every month, and it's hard. You know, I hear statistics and I get kind of blind, but both, we're actually doing this simultaneously. There's an EMA, European submission, and so the idea is that the EMA is very interested, and they have a little bit of a different process for submission. So they're going to get their regulatory submission earlier than the one for FDA, but the responses they get from that will affect that. And this should happen by the end of this year. So we will have step 2 for the FDA done before the year is out, and anticipate a response and then a rebuttal to that sometime in 2022. So we're hoping that by mid to late 2022, we'll have a new regulatory endpoint. How does this affect you? This is my last slide. This is a better pathway for investigator-initiated trials. I mean, even though you're not doing an FDA trial, if you want to focus on late graft failure and you have some cockamamie idea about a therapeutic intervention that look good in animals and you think is safe and you want to try it, or if you want to do an off-label targeting therapy like metformin to treat that, this would be a way to sort of measure that outcome, and maybe more effectively than using 3,000 patients. It does allow open discussions about drug safety and patient-reported outcomes, and that's our other mission in the consortium. It may really help a conversation to help for the clinical validation of current biomarkers. I think that many of us feel that that is really important to have the clinical validation rather than just asking Medicare to pay for it. And that's not a long-term solution, the CMS approval. And also for you all, that there's opportunities for voluntary participation. I mean, you have to be pretty established in the field to do that, but you may have colleagues in your center. Like when I heard about AKI, I reached out to Chirag Parikh and said, dude, you should get involved in this. And he said, I am already. So, but again, it's another opportunity for networking. So with that, my last slide is just to show all the people that were involved both in helping me develop and get the TTC launched, although I can't take credit because it really ended up with the AST and ASTS, but my colleagues at TTS for supporting it as well. Thank you.